Now I would like to invite Miss Sadia Masood, who is an MPhil scholar.
Assalamu alaikum and hello everyone. This is Sadia Masood from National University of Sciences and Technology, and my topic of presentation is preparation and characterization of E. coli DH5 alpha bacterial ghost and their evaluation as a drug delivery vehicle. These are the table of contents which we will discuss in this presentation today. Starting with the introduction, as you all know that cancer is the rapid creation of abnormal cells that grow beyond their usual boundaries, and they can invade adjoining parts of the body and spread to the other organs. This process is known as metastasis, and metastases are the primary cause of death from cancer. These are global cancer statistics for the year of 2020. 19.3 million new cases have been reported for 36 cancers in 185 countries, and 9.9 million deaths have been reported in both genders. So cancer is the second leading cause of death globally, accounting for an estimated 1 in 6 deaths. Talking about cancer treatment and their limitations, the most common treatments are surgery, chemotherapy, and radiations. Other options include targeted therapy, immunotherapy, hormonal therapy, etc. But the most used treatment is chemotherapy, which refers to the use of drugs to kill cancer cells. Chemotherapy has its own limitations, which includes damage to healthy cells, their poor bioavailability. They are not very specific in targeting, and they require high dose requirements. To overcome these limitations, we use drug delivery vehicles to deliver chemotherapeutic drugs to tumor cells. And importance or benefit of using drug delivery vehicle includes their wide damage to normal cells. They are very specific in targeting and reduce the drug side effects and minimize the administration frequency of drugs. The chemotherapeutic drug that we use in our study was doxorubicin, which was one of the most used drugs against a wide range of cancers. And to deliver doxorubicin, the delivery vehicle that we used in our study was bacterial ghost. Now the question is, what are bacterial ghosts? Bacterial ghosts are devoid cell envelopes derived from gram-negative bacteria with intact cellular morphology, and they are advanced system for the delivery of vaccines, drugs, or active substances. This is the graphical representation of bacterial ghost. When bacterial cells releases its cytoplasmic content through membrane tunnels, it will be called bacterial ghost. Talking about the applications of bacterial ghost, there are many applications of bacterial ghost in white biotechnology. They are being used as candidate vaccines against many infectious diseases like Haemophilus influenza. They are being used as adjuvants in vaccines because of their natural intrinsic properties, and they are also being used as delivery vehicle for the delivery of vaccines, drugs, or active substances. We use them as delivery vehicle in our study. Now the question is, why bacterial ghost as delivery vehicle? And the answer to this question is because they are cost effective, they have long shelf life with minimal side effects, and because of their bioavailability, and no cold chain is required as they are in freeze dried form, and because of their easy production and packaging. Keeping in view the above mentioned fact, the main aim of our study was to develop a safe, effective, and targeted drug delivery system for cancer treatment at low cost. And the objectives of our study were preparation of bacterial ghost, and the second objective was loading of doxorubicin into bacterial ghost, and the third objective was release profiling of loaded bacterial ghost. Methodology of our study was, the first step was preparation of bacterial ghost. For this purpose, bacterial strain was revived, and then bacterial ghost was prepared using tween 80 method. After the successful preparation of bacterial ghost, we moved towards the characterization of bacterial ghost. For this purpose, we performed multiple confirmatory tests, which include viability testing, pore size detection, DNA degradation confirmation, and the quantification of released DNA and protein. After the successful preparation and characterization of bacterial ghost, the third step was loading of bacterial ghost with doxorubicin, which is a anti-cancerous drug. For this purpose, freeze-dried BGs were mixed with the drug solution and their loading was confirmed by lysis method. And to calculate entrapment efficacy, we used direct method. After the successful loading of doxorubicin into bacterial ghost, we moved towards the drug release profiling of loaded BGs via dialysis tubing. Samples were taken out at predetermined time periods and then their spectrophotometric analyses were performed and all the collected data were analyzed using GraphPad Prism. Now the results of our experimental work. This graph shows the growth rate of between 80 treated and untreated bacterial cells. As you can see that the untreated bacterial cells were high in growth as compared to the treated bacterial cells with an OD of 0.2 to 0.3, which indicates that E. coli cultures with OD values above 0.4 showed inefficient lysis process, while cultures with OD values between 0.2 to 0.3 resulted in highly efficient bacterial ghost production. For viability testing of E. coli, we performed subculturing, and as you can see, very good growth of untreated bacterial cells on N-agar plates, while the cells which were treated with between 80 showed no growth on N-agar plates even after the incubation period of 3 days. So it concludes that bacterial cells showed no growth on N-agar plates because they were inactivated. We optimized the proposed protocol according to our bacterial strain and then confirmed it by quantification of released DNA. For this purpose, we performed multiple experiments. In experiment A, 200 microliter of inoculum were added into 5 ml of LB containing 7% of twin 80 and the pH of media was 3.6 and the incubation temperature was 37, but the incubation time period was different and the maximum release of DNA was observed at 1 hour standing period, which was 1598.32 nanogram per microliter. And in experimenter B, different volumes of LB containing 7% of twin 80 were used, but the inoculum and pH of media, incubation temperature, and time period were kept constant, and the maximum release of DNA was observed with 5 ml of LB containing 7% of twin 80, which was 15 per microliter. So this concludes that the amount of released DNA was significantly changed in different conditions and the maximum DNA was released after 1 hour standing period at pH 3.6 with 5 ml volume of LB containing 7% between 18 to 200 microliter, which is the optimized condition of bacterial ghost production.